like to short present my work, which is focused on the investigation of baicalein and chitosan effect on biofilm of yeast. The main aim of this study was the evaluation of the effect of amphotericin B as an antibiotic control in comparison with natural substances baicalein and chitosan on Candida parapsilosis, Candida krusei, and Trichosporon cutaneum biofilm. And to the best of our knowledge, this is the first time of using CellaVista automatic microscope with software for image analysis for this evaluation. Biofilms are highly organized matrix enclosed microbial communities which are irreversibly attached to the surface interface or to each others. And these microbial communities are able to cause a wide range of human infections. Candida species are one of the most often isolated yeast from nosocomial infections. They can cause, for example, oral infections, endocarditis, ocular infections, or joint diseases. And also Trichosporon cutaneum can cause some infection, and it is dangerous, especially for immunodeficient patients. And these filamentous yeast can cause superficial biofilm-associated infection like onychomycosis. The huge problem of these infections is the high antimicrobial resistance against typically used antibiotics, for example, amphotericin B. And therefore, the attention is turning to the use of natural substances with antibacterial, antifungal, or anti-biofilm activities, because these substances can be used in low concentration and no resistance against them is developed. So the examples of these substances are baicalein and chitosan. Baicalein is flavonoid which is obtained from Scutellaria baicaleensis, concretely its roots, and it has antibacterial, neuroprotective, antiviral, and antifungal activities. And the second one, chitosan, it is present in the outer shell of crustaceans, and it has also a broad antimicrobial properties. The Candida strains used in this study were kindly provided by a collection of microorganisms UCT Prague, and Trichosporon cutaneum was obtained from Slovak Academy of Science. And these yeasts were cultivated in 96 wells polystyrene microtetraplates, similar like this one. And the colonized area of the bottom of the wells was evaluated by CellaVista. CellaVista is an automatic microscope with software for image analysis, and it brings new possibilities in qualification and quantification of the sessile cells or biofilm in the same time. On this picture, you can see the evaluation process. Image A is depicting the sessile cells, and image B is a binary image where the black area means the colonized area of the bottom of the wells. Using these methods, we investigated the effect on microbial adhesion and also the effect of these selected substances on eradication of preformed biofilm. To test the inhibition of microbial adhesion, the yeasts were cultivated in presence of selected substances from the very beginning. And to test the eradication of preformed biofilms, the yeasts were cultivated and after 24 hours in the case of Candida species and 48 hours of cultivation in the case of Trichosporon cutaneum. Each well was 3 times washed by saline, and at the appropriate concentration of either one of the natural substances or antibiotic were added to the wells. And after 24 hours of cultivation, each well was washed, and the plate was evaluated by Cella Vista. Now you can see the final results. In this table are shown the minimum inhibitory concentrations. These concentrations were our starting concentrations for finding the effective concentration for inhibition of much more resistant sessile cells in biofilm. In this case is the important one, the blue one, 1.5 milligram per liter of amphotericin B, which inhibits the growth of suspension cells of Candida parapsilosis. And in this figure you can see that amphotericin B is able to inhibit the microbial adhesion in concentration 3 milligrams per liter, which is 2 times higher than MIC, but it promotes the biofilm formation instead of eradicating it. On the right side, there are binary images which depicting the effect of amphotericin on microbial adhesion or on eradication of preformed biofilm of Candida parapsilosis. In the case of Candida krusei, this antibioticum was able to reduce the biofilm formation of Candida krusei, but there was no significant effect on inhibition of microbial adhesion. But Trichosporon cutaneum is not absolutely sensitive to the amphotericin B effect or action. On binary images, you can see that biofilm structure of Trichosporon cutaneum is different. It is because its capability to form pseudomycelium in suspension also in biofilm. And the interesting question for us was the effect of natural substances. In this case, it was baicalein. And now we can see that baicalein is able to reduce the biofilm formation of Candida parapsilosis even in concentration 20 milligram per liter, which is more than 10 times lower than MIC, which is really interesting result. But it has no significant activity against microbial adhesion. The similar results were obtained in the case of Candida krusei, but Trichosporon cutaneum is extremely sensitive to the baicalein action. In comparison with antibiotic amphotericin B, it is a really important result. And the second natural substance is chitosan, is able to inhibit microbial adhesion, but it has no significant effect on eradication of biofilm of Candida parapsilosis. In the case of Candida krusei, it is able to inhibit microbial adhesion in the lower concentrations and also hasn't got any significant effect on eradication of biofilm, and the similar effect was confirmed in the case of Trichosporon cutaneum. So in conclusion, amphotericin B is not able to eradicate the sessile cells of Candida parapsilosis and Trichosporon cutaneum, contrary to baicalein, which has the ability to reduce preformed biofilm of all tested yeast strains, and its antibiofilm activity is in lower concentration than its antimicrobial activity against suspension cells. Chitosan is able to significantly decrease microbial adhesion, but hasn't got any significant effect on eradication of biofilm. And our plan to future, we would like to find a synergistic effect of these selected substances in combination on biofilm formation inhibition. So I would like to thank my supervisors and my colleagues, and also thank you for your kind attention.